Welcome back to this next video in which we are studying headaches. Okay, so we are currently in the process of discussing the anatomy of the sensory nucleus of the trigeminal nerve. And remember, we have seen that this is an enormous great big nucleus that spans throughout the brainstem and even into upper regions of the spinal cord, specifically the cervical levels C1 and C2 of the spinal cord. So because this nucleus is so large, we usually split it into three separate parts. The mesencephalic part, which is the bit that stretches up into the midbrain, the mesencephalon. The principal sensory nucleus of the trigeminal nerve, which is this plump bit in the middle of the pons. And then the spinal nucleus of the trigeminal nerve, which is this bit that extends right down through the medulla and then into the cervical levels of the spinal cord below. And to get a complete understanding of the anatomy of this trigeminal nucleus, what we have been doing is taking cross-sections through at different levels to see where it is relative to other important structures at that level. So far we have taken a cross-section, an axial section through the rostral midbrain and seen the mesencephalic portion of the sensory nucleus of the trigeminal nerve. We've also taken a section through the middle of the pons and therefore saw the principal component of the sensory nucleus of the trigeminal nerve along with the motor nucleus of the trigeminal nerve, which is a far more simple shape to understand. Now what I want to do is take a cross-section through the medulla and then we'll go on to taking a cross-section through the cervical spinal cord. So, to begin with then, a cross-section through the medulla. What level of the medulla am I going to take a cross-section through? I'm going to go right in the middle here. This is the level I want to do my cross-section through. And the reason for that is that if we take a cross-section there, we will catch the inferior olives. And also, look over here, we'll catch the dorsal column nuclei. That section is going to be below the inferior cerebellar peduncles, so we won't see the inferior cerebellar peduncles, we won't see the fourth ventricle. Instead, we'll see the four dorsal column nuclei at the back, and we'll also see the central canal of the medulla, which is a continuation of the fourth ventricle. Just like the fourth ventricle above is continuous with the cerebral aqueduct, below it is continuous with the central canal through the medulla, and that's then continuous with the central canal that goes through the cervical level of the spinal cord. So the reason I'm taking this cross-section overall in conclusion is that we will see the inferior olives, we will see the dorsal column nuclei, and we will see the central canal of the medulla. Later on, we will also discuss what the higher up cross-section where you see the inferior cerebellar peduncles would look like when we come on to discuss area postrema, when we want to discuss nausea and vomiting as an accompanying symptom of headaches. But that's later on. So, let's begin with this cross-section. So, let's go over here, I think, and let's draw its cross-section. So, let me decide on a colour. We'll have this pinky colour. So, we'll stick to the format that we've been using before, where we draw the back at the top and the front at the bottom. So, the back then, the back is going to have these four protuberances, which are going to be the dorsal column nuclei, very famous structures amongst medical students at least. So, let me put these in. So, there is one dorsal column nucleus, there is another, here's another, and here is the final one. So again, remember, we are looking from above. We're imagining we're looking from above, so this is the right side and this is the left side. So here are the right dorsal column nuclei and here are the left dorsal column nuclei. So let's extend it forward. How is it going to work as we go forward? Well, at the front of the medulla, we know we have the pyramidal tracts, the pyramids of the medulla, and also, of course, behind them, the inferior olives. So let's try and mark this on. So here we go, it comes forward, we'll have the inferior olive here, and then we'll have the right pyramid here, the left pyramid here, and then the left inferior olive will be in this position here. Now, my cross-section of the medulla has ended up looking very square-like. It's not quite as square as that, it's a little bit more round. However, this is a good enough picture for us. So, let's start marking on some important structures then. So, first one, right at the front of the medulla, we have the pyramids, which are the pyramidal tracts, the corticospinal tracts. So these have come down through the cerebral peduncles. They are axons coming down from the primary motor cortex, where the cell bodies of these that these axons belong to are. They've come right down through the cerebral peduncles, they've gone through the pons, and they're now going through the medulla, and here they are making these protuberances on the front of the medulla here. So in blue, these are the two corticospinal tracts, and for short I'll abbreviate corticospinal tracts to CSTs. So there are the corticospinal tracts. Next things to add on are the things just behind the corticospinal tracts, which are the inferior olives. Now, these are not missable when you take a cross-section of the medulla, unless you take the cross-section too low, then of course they won't be there. But these are massive, great structures. If you take the cross-section of the medulla at the right level, you cannot miss the inferior olivary nuclei. They are enormous, great things. So let me put them on here. And they don't just make up that bit that's actually visible on the front of the medulla, which we call the inferior olive. They also go behind the corticospinal tracts like so. So there is the right one, and here is, the left inferior olive. So those in grey there, those are the inferior olivary nuclei. We'll just refer to them as the inferior olives. So, in case you don't know what the inferior olives do, let me just give you a tiny bit of background. It's not relevant to headaches. It's another part of the cerebellum, actually, or the cerebellum function. So I've told you about how the cerebellum is very important in calibrating movements that need to be precisely calibrated, and the, archetypal example of that is catching a ball that needs to be calibrated to visual information. How does the cerebellum learn to calibrate movements precisely? You are not born knowing how to perfectly catch balls. It's something that you learn. The cerebellum has to learn to perfectly calibrate these movements. How does it learn? Well, we don't know exactly, but a current theory believes that the inferior olive is extremely important in helping the cerebellum to learn how to calibrate movements precisely. So the inferior olive, it's important in cerebellum motor learning, is the nice one-line answer you can give if someone asks you what the inferior olive is involved in doing. Okay, next thing I think to put on is probably the dorsal column nuclei, very famous structures amongst medical students. Like the corticospinal tracts, which are very famous among medical students, the dorsal column nuclei are probably the second most famous things. So, you have 4 dorsal column nuclei that are symmetric on each side, so we'll start with these medial ones here. So these are the gracile nuclei, that's the plural. The singular that I prefer is nucleus gracilis. So if I was just talking about one of them, for instance the left one, I will call it the left nucleus gracilis. And I believe gracilis only has a single L in. If I wanted to use the plural, I would refer to them as the gracile nuclei, rather than nuclei gracilis. How would you work it out? No, I don't know. I'd refer to it as the gracile nuclei. So you have one on both sides, the left nucleus gracilis and the right nucleus gracilis. Now let's do the one to the side of it. So what colour should we go to now? I think we'll go to red. Let me get another more interesting red here, the evil red for some reason I've come up with for this one. So here is the one to the side of it, the other 2 dorsal column nuclei. These are the cuneate nuclei. Again, the singular that I prefer is nucleus cuneatus. So we have a left nucleus cuneatus and a right nucleus cuneatus, and the plural for that is to call them the cuneate nuclei. So these are the dorsal column nuclei. They are part of the very famous pathway that all medical students learn, which is the dorsal column medial lemniscal pathway, which is the pathway by which fine touch sensation, proprioceptive sensation, and vibration sensation, those 3 modalities of general sensation, it's the pathway by which they get up to the primary somatosensory cortex, and we'll go over that more later on. These nuclei contain a synapse between the primary neuron that has sent its central process all the way up the spinal cord in the dorsal columns to these nuclei, and the second order neuron that will be a multipolar neuron with lots of dendrites that will be where the central process axon is synapsing onto. And they will send their axons up then to the thalamus, or the thalami rather. And they're going to send their axons up to the thalami through the medial lemnisci. So the medial lemnisci are part of this pathway. So just to write on the fact that these are the dorsal column nuclei, both of these. So all four of these, in fact, but both of these types of dorsal column nuclei. So these are the dorsal column nuclei. They're the nuclei at the top of the dorsal column, and they're part of this dorsal column medial lemniscal pathway, which for short is abbreviated to the DCML pathway, dorsal column and then medial lemniscus pathway. Right, so the next thing, of course, to add on is probably the medial lemnisci. Actually, before we do the medial lemnisci, let me put on the central canal of the medulla. So remember, this is continuous with the fourth ventricle above, and this is right in the middle in this sort of position here. So that is the central canal of the medulla containing CSF, and it's going to be continuous below with the central canal of the spinal cord as well. Okay, next up, let's put on the medial lemnisci. So this is a bit confusing because you might expect, if you didn't know better, that the medial lemnisci would be in this sort of position. That would be the natural guess, because look at the other pictures we've drawn. Here has been the medial lemnisci in the pons, and here are the medial lemnisci in the midbrain. And we can nicely imagine how you can go up from the pons to the midbrain and end up with them at this sort of angle. We can nicely imagine how they're continuous with one another. Now, on this new picture that I'm going to draw here, the medial lemnisci are going to be on a completely different angle, at right angles to the way they've been oriented previously. So let me put them on here. So they are in this position, like so. So there is the right medial lemniscus. I had to think for a moment there. And here is the left medial lemniscus there. So both of those structures, they are the medial lemnisci, which I'll just abbreviate to ML for short. Okay, so how do they go to being on this total different orientation here? Well, as they go up into the pons, they twist around, and the way they twist is this comes forward, like so, and this kind of goes backwards a little bit, so that it ends up twisted like this. But this bit is continuous with this, and this side here is continuous with this. That's the way it twists, rather than twisting the other way round. So it twists this way round to become like this, on this orientation. So the medial lemnisci are slightly more complicated. They're not just flat all the way along. Instead, they are twisting at that junction between the medulla and the pons. Okay, now, coming back to the dorsal column medial lemniscal pathway, which we will summarize later on as we've seen, once we've seen the anatomy of the cervical spinal cord. And of course, later on we're going to discuss how does sensation from the back of the head and the neck region actually get to the brain, and that will require us to discuss the dorsal column medial lemniscal pathway. However, to justify a structure that I'm about to put on here, I need you to know a little bit more about the dorsal column medial lemniscal pathway. So hopefully this is revision, but remember, when we have our primary sensory afferent on one side of the body, let's say, just to take a specific example, let's say it's on the right side of the body. So we've got a primary sensory afferent of one of the modalities that is going to go up the dorsal column medial lemniscal pathway, and I'll remind you that's fine touch, vibration, and proprioception. So it's come in, it's taken some sensation from the right side of the body. It will have its cell body in a dorsal column, not in the dorsal column nucleus, in a dorsal root ganglion. Its central process will then enter the spinal cord. It will go up the dorsal column of white matter, which we'll see in just a moment when we go on to the cross-section of the spinal cord. It will send up in the dorsal white matter column, and that's important to understand. The central process is very long. It's going to go right up the spinal cord, it will come up into the medulla, and then it will synapse in one of these two dorsal column nuclei on its side. So it will a synapse in either the right nucleus cuneatus or the right nucleus gracilis. And generally, the lower down it comes from the body, the more likely it will be to synapse in the nucleus gracilis. And if it's higher up the body, maybe it's come from the arm, it's more likely to synapse in the nucleus cuneatus. So gracilis mediates the lower body, cuneatus is for the higher up regions in the body. So indeed, sensation of these modalities from the head and neck region, back of the head and neck region, would come up and synapse in the cuneate nucleus rather than the gracile nucleus. And you can use these terms, as I just have, for singular as well. You can call the nucleus gracilis the gracile nucleus, and you can call the nucleus cuneatus the cuneate nucleus. So at the moment, everything's remained ipsilateral, but we know that the left cerebral hemisphere mediates the right side of the body, and the right cerebral hemisphere controls the left side of the body, and that applies to somatosensation as well. So we need to cross over, and this is where we're going to cross over. The second order neurons that have their cell body in these dorsal column nuclei, they're going to send their axons into the medial lemniscus, but they're going to send them into the contralateral medial lemniscus. So if we're talking about the right dorsal column nuclei, they're going to send their axons into the left medial lemniscus, which will send up to the left thalamus, which will then go up to the left primary somatosensory cortex. So, what you then see in this cross-section of the medulla is you will see the fibres crossing over, because loads of them are crossing over. So many are crossing over, you can actually see lines which are representing loads of fibres moving over. So let me try and draw this. So from the right-hand side first, here come fibres crossing over into the contralateral medial lemniscus, the left medial lemniscus here. Okay, and now I'll do it on the other side. So here we go. Here are fibres coming over from the left dorsal column nuclei into the right medial lemniscus. So you have this crossing over as part of the dorsal column medial lemniscal pathway. And those fibres that you can see, you can get an impression of them in a cross-section of the medulla. They are called the arcuate fibres, so they are a key landmark to be aware of when you're looking at a cross-section of a medulla at this level. Okay, so the arcuate fibres, part of the dorsal column medial lemniscal pathway, they are the axons of the second order neurons crossing over into the contralateral medial lemniscus to ascend to the contralateral ventroposterolateral nucleus of the thalamus. Okay, so next things that we need to put in. I will put in the reticular formation in the medullary level first, and then we'll put in the spinal nucleus of the trigeminal nerve and the spinal tract of the trigeminal nerve. So reticular formation, I'll go for a grey, actually no, we've already done grey colour, we'll go for the bluey colour. So in this region here is where the medullary reticular formation will be. And remember, the reticular formation, you have only one of them. You don't talk about having a left and a right reticular formation. It's on both sides and it's throughout the brainstem. This is the bit that we're seeing in the medullary level. It will be continuous with the reticular formation up in the pontine level, which will then be continuous with the reticular formation up in the midbrain level. So I'll label it up. This is the reticular formation in that colour. And hopefully this is all starting to make sense. Look at this picture that we had over here of the pons. Here's the reticular formation, and we know that that's going to be continuous with the reticular formation here of the medulla. Okay, and now hopefully you can guess where the spinal nucleus of the trigeminal nerve is going to be. The principal sensory nucleus was in front of the reticular formation and very lateral, so indeed the spinal nucleus of the trigeminal nerve is also going to be in front of the reticular formation and very lateral, so it's going to be in this sort of position here. So everything does sort of make sense, hopefully. So let's get vivid purple back again, and remember we're not just putting on the spinal nucleus, we're putting on the spinal tract, and the spinal tract is lateral to the spinal nucleus. So let me start by putting on the spinal nucleus. So it's on the right hand side, and here's it on the left hand side. So that is the spinal nucleus, and I'll use N for nucleus as well, of the trigeminal nerve. So N is either nerve or nucleus. So that's the spinal nucleus of the trigeminal nerve's position, where the cell bodies of the second order neurons for the trigeminal system are going to be, and they're going to send their fibers not into the medial lemnisci, but into something called the trigeminothalamic tract, which sometimes is called the trigeminal lemniscus, which we'll discuss later on. And then lateral to it, which I'll put in white here because it is a white matter tract, is the spinal tract of the trigeminal nerve. So in white there, that is the spinal tract of the trigeminal nerve. And let me just remind you of the intuition behind what the spinal tract of 5 is. The central process of the pseudo-unipolar primary sensory afferent that was in the trigeminal nerve comes in at the pons level through the trigeminal nerve. So going back to this picture, it's coming in through here. If it was going straight into the principal sensory nucleus, then it would have a very short journey. It would go straight in there, synapse onto a second order neuron. Fantastic. But what if it's coming down all the way here? What if it's going right the way down here? Then its fibre will have to go all the way down like this. And you do have a significant number of fibres coming to the spinal nucleus of the trigeminal nerve. So when you take a cross-section through here, you do see a significant white matter tract there. You see a significant piece of white matter, and that is the fibres running down to lower levels of the spinal nucleus of the trigeminal nerve. So that's what the spinal tract of the trigeminal nerve is. Excellent, and that is pretty much the picture of the medulla now completed. So the next thing we want to do is do the cross-section of the cervical spinal cord so that I can show you where the where the spinal nucleus of the trigeminal nerve is in the grey matter of the spinal cord. And I'll give it away, it's going to be in substantia gelatinosa. Which is also happens to be where the nuclei of, where the cell bodies of second order neurons of the spinothalamic tract that will mediate nociception of the back of the head and neck region will also be. Therefore, it's all kind of working together, and that's where the concept of the trigeminal cervical complex comes from. But we'll paint that picture later on. Okay, so for now what I want to do is remind you of what a cross-section of the spinal cord looks like. Now, you know, if you take cross-sections of the spinal cord at different levels along the spinal cord, it does look slightly different. However, it's the same basic picture. If you do it between T1 and L2, you have to add on an additional grey matter horn. You have to remember to put in the intermediate grey matter horn, which is where the cell bodies of the preganglionic sympathetic fibres will be located. But apart from that, there isn't a major difference in what a cross-section of the spinal cord will look like. So this picture that I'm about to draw you, it's actually really the same picture I'd use for most levels of the spinal cord, not just the cervical levels, but of course we are drawing it to understand the cervical levels. So let me start by drawing the outline for it then, and I'll try and get this as symmetrical as I can. So like so, we will have at the front the anterior median fissure of the spinal cord, and then we'll come back, and at the back we have the posterior median sulcus, and I'll try and get this opposite the anterior median fissure. So remember, a fissure is an actual open space, whereas a sulcus is a potential space. Potentially you could open a space here, but it isn't usually actually open. So let me just put those names down. So we have the anterior, which I'll abbreviate to ant, median because it's in the middle, and it's the fissure, and that runs all the way down the spinal cord. So right the way down the spinal cord you have a fissure right at the front, which is the anterior median fissure. And let me just remove that horrible splodge that I've managed to do. And then at the back, of course, we have the posterior median sulcus. So this is not an actual open space. Instead, that is a potential space that you could open up. It's usually actually closed. So that's the posterior median sulcus. So those are just some landmarks of a cross-section of the spinal cord, the anterior median fissure and the posterior median sulcus. Next thing to do is mark on the grey matter of the spinal cord, and then we'll also have the white matter of the spinal cord on there as well. So remember, grey matter is where you're going to have cell bodies of neurons, and white matter is where you're going to have axons of neurons. So the grey matter of the spinal cord, it has this sort of butterfly shape. You have the dorsal horns and the ventral horns on both sides, and if we were in between T1 and L2 level, we'd also have to put the intermediate horn on there, which is where, as I said earlier, the preganglionic sympathetic fibers would have their cell bodies, and those would come out in the mixed spinal nerves between the levels of T1 and L2, and then go into the sympathetic chains. Okay, so let's mark on the grey matter then, and we're doing cervical level, so we don't need to worry about the intermediate horn. So here we go. So we're starting with the dorsal horns here, so there is the left dorsal horn, and here is the right dorsal horn, and then at the front we'll have the ventral horns, like so, and I'll try and get it as symmetrical as possible. Whoops, made a horrible mess there. Let me just sort of try and get rid of that bit there. So let's try and make that a bit smoother. There we go. It's not quite symmetrical, but it's okay. Actually, I'll try and bring this forward a little bit more so it is closer to being symmetrical. There we go. So those are the two ventral horns. So grey matter of the spinal cord is split into the dorsal horns, the horns at the back, and the ventral horns, these horns at the front. So we call them the dorsal grey matter horns and ventral grey matter horns. And then in the middle of the grey matter, you'll then have the central canal of the spinal cord here. Other landmarks other key pieces of terminology that we'll just revise, and which I'm hoping you are familiar with, are the names for the different white matter columns. So all around us is white matter. This is white matter, this is white matter, this is white matter, this is white matter, this is white matter. You know, all of this is white matter. And we split the white matter up. These are the dorsal columns here, so these are where the axons of those central processes of the pseudo-unipolar primary sensory afferents of the modalities fine touch, proprioception and vibration will ascend up to get to the dorsal column nuclei above them in the medulla as part of the dorsal column medial lemniscal pathway. Then this bit of white matter that's behind the dorsal horn of grey matter, that has a special name. That is called the Tract of Lissauers. So let me just mark this on in aquamarine colour. So this piece of white matter that I'm separating out, because this is a name that you might not have heard of, this is called the Tract of Lissauers. And when, for instance, in sensory neurons, when the central process of the pseudo-unipolar primary sensory afferent comes into the spinal cord through the dorsal root, it can move in the tract of Lissauers to get to a slightly different level. So the dorsal root of a mixed spinal nerve, which is where all of the sensory fibers are coming in, it's going to come in as a little level of the spinal cord, but you don't want all of the central processes to go and synapse right at that level necessarily. So they can move up and down a little bit in the spinal cord through this tract of Lissauers. So this is where central processes of pseudo-unipolar primary sensory afferents that are coming into the spinal cord can move up and down. And indeed, this is what is continuous with the spinal tract of the trigeminal nerve. This is white matter fibers. So it's good that we are stressing that. I am stressing that for an important reason. So that's the Tract of Lissauers. Let's do names of more white matter columns. This is the lateral white matter column, and of course you have the right lateral white matter column here as well. These are the anterior white matter columns, so left anterior white matter column and right anterior white matter column. And then in front of the ventral horns here, those are the anterolateral white matter columns. So those are the names of the different white matter columns. Now, bits that we want to see, of course, we want to see where the spinal nucleus of the trigeminal nerve is. I've already told you where the spinal tract of the trigeminal nerve is. So fibers from the trigeminal nerve, the central processes of the pseudo-unipolar neurons from the trigeminal nerve that are coming right down into the cervical levels of the spinal cord before they're going to synapse, they will be coming down in the tract of the Zowers. So it's continuous with the tract of the trigeminal nerve, the spinal tract of the trigeminal nerve above in the medulla. Okay, where is the actual equivalent of the spinal nucleus of the trigeminal nerve then? It's in the rexed laminae number 2, which is also called the substantia gelatinosa. So, I'm going to just briefly remind you of the division of the grey matter of the spinal cord up into rexed laminae. So there is a division of all of the grey matter up into these laminae called the rexed laminae. And there are overall 10 rexed laminae, and we number them with Roman numerals because it's an old categorization and people like to be pretentious and use Roman numerals. So, the first 6 rexed laminae are in the dorsal horn. They're parts of the dorsal horn. So let me do it just on one side. So there's the first one, second, third, fourth, fifth, and 6th. So, just to highlight this again, that's the first one. Here's the second one, third one, fourth one, fifth one, 6th one. So they are rexed laminae, number 1, the Roman numeral for number 1 there, to number 6. Okay, so those are the first 6 rexed laminae. I'm also just going to put a division of the grey matter into the 2 halves. So I'll put a division down here, because of course this is the left grey matter and this is the right grey matter. So those are the first 6 rexed laminae. Now for the final 4 rexed laminae. So I want to put on 10 first, because these ones are a little bit more complicated to actually show. So I'll put on 10 first. 10 is this sort of region here. So that is rexed laminae number 10, there in green. Now let's put on rexed laminae number 8, which is just in front of rexed laminae number 10. So this one here in blue, that is rexed laminae number 8 there. And then rexed laminae number 9 is not simply connected, to use the fancy maths word. What this means is it's not just one blob, it's 2 blobs. So the region that is rexed laminae number 9 is those 2 blobs, this blob here and this blob here, that aren't actually simply connected to one another. They're not connected, they're separate. Okay, so that is rexed laminae number 9 there. And just to give you some other background knowledge about the rexed laminae number 9 is where the motor neurons that come out of that ventral horn are going to have their cell bodies. So that's the definition of rexed laminae number 9. It's where the motor neuron cell bodies are. So remember, throughout the spinal cord, and these rexed laminae, by the way, you can apply these to the grey matter of the spinal cord all the way along the spinal cord, not just to the cervical level, and all the way along you're going to have motor neurons which are multipolar neurons with their cell bodies sitting in the ventral horn, sending their axons out through the ventral root of the of a mixed spinal nerve, and they're in this rexed laminae number 9. Rexed laminae number 10 has another name. It's also called the grey commissure. So here's another bit of background knowledge. Rexed laminae number 10 is also called the grey matter commissure. Rexed laminae number 7 is the one I haven't given you yet. Rexed laminae number 7 is the one we have to leave till last, because it's everything that's left over. All the rest of the grey matter, all of this that I'm colouring now in aquamarine there, that is rexed laminae number 7, which has another name. It's also known as zona intermedia. So that's zona intermedia, the intermediate zone. Zona intermedia. Now, there are also other names for the different parts of the rexed laminae 1 to 6. So rexed laminae number 1 is also called the marginal zone of the dorsal horn. So 1 is also called marginal zone. And the important one for us to know is actually the other name for rexidlamine number 2, because rexidlamine number 2 is also called the substantia gelatinosa. Now, why am I so keen on this one? Because this is actually the continuation, effectively, of the spinal nucleus of the trigeminal nerve. This is where second-order neurons that are part of the spinal nucleus of the trigeminal nerve that's actually in the cervical regions of the spinal cord will have their cell bodies. So this is substantia gelatinosa, rexidlamine number 2. And just for some more background, let's just continue on. So 3 and 4 together, so rexidlamine number 3 and rexidlamine number 4, together they are called the nucleus proprius. So 3 and 4 are the nucleus proprius. And finally, rexidlamine number 5 and rexidlamine number 6 together, they are called the neck of the dorsal horn. So, neck of dorsal horn. Right, so there we go. Those are other names for the 10 rexidlamine. So, that is how we divide grey matter up throughout the spinal cord, not just in the cervical region. And the bit that we are really interested in is this substantia gelatinosa, rexidlamine number 2. So overall, the spinal tract of the trigeminal nerve, this is continuous with the substantia gelatinosa of the first two cervical levels, C1 and C2. The substantia gelatinosa is the continuation of the spinal nucleus of the trigeminal nerve. So, if you are a primary sensory afferent in the trigeminal nerve that is going to send its central process down into the portion of the spinal nucleus of the trigeminal nerve that's in the C1, C2 level, your central process will go down through the tract of the Zowers. Well, first it will go through the spinal tract of the trigeminal nerve, through the bottom of the pons, through the medulla, and then it will go into the tract of the Zowers, which is the continuation of the spinal tract of the trigeminal nerve, and then it will synapse on the second-order neuron that will have its cell body in substantia gelatinosa, which is the continuation of the spinal nucleus of the trigeminal nerve into the C1 and C2 levels. Okay, so we'll have a break here. In the next video, we'll have a look at where the axons of these second-order neurons in the trigeminal sensory nucleus are actually going to go, and that's the trigeminal thalamic tract.